We seem to have slowed adding participants a little bit. Nathan, are you feeling ready to get going?
Yeah, I'm good to go.
Okay. Well, we'll go ahead and get started. So I'm Mark Ducey. I'm Nathan's Thesis Committee Chair, and welcome to everybody. It's really great to see a lot of friends, colleagues, people from the Forest Service, members of the conservation community who have joined us here, and that's testimony, I think, to the relevance of what Nathan's been doing. It's been a really fun project, building on some work that Andy Coulter and Olivia Fraser and others were involved in on the White Mountain National Forest. And so there's a lot of thanks to go out to people who helped get started and give Nathan a great springboard to jump off from. And the other person, really a lot of this is Tom Lee's fault. I still remember, well, it's coming up on about 3 years ago, and it's this time of year, and start to think about taking on new grad students, and I get contacted by this former UNH student, but not one that I had met somehow, and get this application. It looks really good. I talk to this guy, and he's really intense. I'm like, Tom, this student said he worked with you. I mean, what can you tell me about him? I mean, he's really yeah, I mean, really intense. And Tom's like, yeah, you should take him. I'm like, okay, done. So that sort of started things off in what has turned out to be for me at least, an absolutely fantastic direction. Nathan's picked up some of the work that Andy had started, added data. I see Joanne Hoy was on the call as well here, with some, and Emily, and with some help in the field from others. And he's also really thrown himself very seriously into a pretty analytical area of community ecology, and has some conclusions to tell us about today. So with that, I'm going to turn it over to Nathan and let him tell you what he's been up to for the last couple of years.
All right. Thanks, Mark. I guess one thing I'll say just to start off is it might be useful just to see the whole slides if you put just kind of like one thumbnail up in the top right corner. I tried to design most of my slides so that if you have a small thumbnail up at the top, you can still see some gestures that I might make, but not miss out on any of the content of the slides. But yeah, so with that, yeah, I'm Nathan Roe, and I'll be talking about my master's research. So the topic is how species distribution models can improve our understanding of plant spatial patterns in the White Mountains of New Hampshire. And so I'll be easing into this topic of species distribution models in the next few slides. But in essence, I'll be talking about the associations between plants and environmental conditions, and how those associations can help us to predict the location of species, as well as understand the ecology and evolutionary history of these species. So these are really questions related to biogeography. And so this word biogeography is just a combination of biology and geography. So we're looking at the spatial distributions of organisms. So if you look at the photo in the back left, an example of a biogeographical question would be, why is pink lady slipper and poison ivy, red oak, hemlock in this site, and why are other species absent from this site? Maybe, for example, sugar maple, baneberry, lady fern. And so these types of questions have been talked about and written about for a couple hundred years, starting with early biogeographers and naturalists. But undoubtedly, people have been asking these questions for far longer, hundreds, if not thousands of years. And so what drives spatial patterns of species, there's several contributing factors. The dispersal ability of species can be a factor. Past land management history or disturbance can be a factor. But one of the major factors is niche. And this term niche was introduced to ecology in the early 1900s by Joseph Grinnell. And the term came for reference, as you can see in the photo to the right here, the area, kind of recessed area where statues occupy. And when Grinnell brought this into ecology, it really refers to the ecological space that species occupy. And I mentioned that early biogeographers and naturalists were thinking about this. So people like Alfred Wallace and Humboldt and Bonpland and Darwin were thinking about the associations between species and environmental conditions. And Grinnell kind of formalized this in the early 1900s. And then several other ecologists have expanded these ideas into what's considered niche theory. That includes G. Evelyn Hutchinson and Robert Whitaker, Robert MacArthur. And I'm going to talk a little bit more about Hutchinson's definition of niche. It's frequently discussed in ecology, and it fits in really nicely with species distribution models. So Hutchinson defined niche as an n-dimensional hypervolume, which is really just to say that the niche of species can be defined with different numbers of variables. So you could have a one-dimensional niche, 5-dimensional niche, 10 dimensions, and so on. And so we'll construct a Hutchinsonian niche to kind of make this this clear. So we could have a first dimension, which could be pH, and a species could have a minimum and maximum threshold to pH. And between these thresholds, the species would be capable of being present. And we could add a second dimension to this species niche, and it could have a minimum and maximum threshold to this dimension light. And this interior area here is where the species would be capable of being present. And so this is a 2-dimensional niche. We could add a third dimension. It could be temperature. A fourth dimension could be related to moisture. And so then you get into these higher dimensional niches. And that's why Hutchinson described it as an n-dimensional hypervolume, something with higher dimensions. So this fits in really well to species distribution models. And species distribution models are basically statistical models that estimate the niche of species and allow you to make predictions about the distribution of species. And they do this using species occurrence data and environmental conditions. So if you look at this picture, we have some wild geranium here. We could look at where wild geranium is present or absent and the environmental conditions where it's present or absent, and then use some statistical techniques to estimate the niche of that species. Within what environmental conditions is this species capable of being present? And so early SDMs really did just this. They were called envelope models, and they basically define the boundaries of kind of a Hutchinsonian niche. So if you have pH and you have a minimum maximum threshold, anywhere in between those, these models consider the species being capable of being present. And the probability of occurrence within those thresholds would be treated as constant. But from work from people like Robert Whitaker in the 1950s, we know that species probably have more complex relationships with environmental gradients. Whitaker talked about in theory how they're likely Gaussian distributions. And so there's a minimum and maximum threshold, but there's likely an optimal range at which point a species abundance is the greatest or the probability of occurrence is the greatest for that species. And so once you have this estimate of niche, there's a couple directions you can go in. One is to explore the niche of that species, as you can kind of see in the lower left here, and we'll be talking a good bit about that. And the other thing is to go a step further. You take the estimate of niche, and then you take known distributions of environmental variables that define that species niche, and that allows you to make predictions of the distribution of these species. And there are two critical assumptions that are inherent to SDMs. One is that species are well dispersed. So if you estimate the niche of a species and you look at a site and it has favorable conditions for that species, the assumption is that the species will be present at that site. And then the other assumption is that all ecologically relevant predictor variables are used when you're estimating the niche of the species. And this assumption has been discussed a lot in SDM literature because it's commonly violated. Most SDMs look at species only in reference to climate variables like temperature and precipitation, and they omit a lot of ecologically relevant predictor variables. For instance, we know for plant species that light and soil conditions are really important to the niche of those species and drive their distributions. And so we're particularly interested in the role of soil variables in SDMs and how important they are. There haven't been that many studies that have looked at the importance of soil variables in SDMs because it's time consuming and expensive to dig soil pits and analyze soil chemistry. So with that, we have a couple of objectives. The first one is to determine the importance of soil variables. As I mentioned, Not too many studies have looked at this. Our soil chemistry data set is as detailed as any other SDM that I've seen. There's only one other study that I'm aware of that has the same level of detail in soil chemistry as what we have. And so this is more of an academic kind of objective to contribute to the body of literature surrounding SDMs. How important are soil variables? And for studies that are omitting soil variables, how much accuracy are you losing? How important is that violation of that assumption? And then the other objective is to actually look at these species that occur in the White Mountains and determine, what environmental gradients are driving their distributions. How could we potentially use these species to predict environmental conditions at these sites? And so before I jump into those objectives, I'm going to go over a quick overview. So these are kind of methodological and study design things that contribute to both of these objectives. So this project is a collaboration between the U.S. Forest Service, the NRCS, and the University of New Hampshire. We worked in the White Mountain National Forest. We had 200 plots and 41 study species. The response variable is the understory vegetation, and the predictors are soils data that includes soil chemistry like exchangeable calcium, also qualitative data like soil texture, LiDAR data. So we flew a plane over the study area, sending lasers down to create a very detailed three-dimensional topographic model of the study area. And then multispectral data using the European Space Agency Sentinel-2 satellite, looking at spectral reflectance of the vegetation, which is associated with the health and vigor of vegetation, as well as some of the chemical constituents of the vegetation. And so in SDMs, predictors are generally broken down into what's called direct and indirect variables. And direct variables are really the gold standard in SDMs. For plants, they include temperature, water, nutrients, like our soil conditions, light, disturbances, and biotic interactions. And the reason that these are the gold standard is that plant fitness directly responds to these variables. So it's easy to interpret how these variables are influencing the distribution of plants. Indirect variables can be really valuable predictors, but they can be a little bit more difficult to interpret. So our spectral data and our topographic data, these are indirect variables. An example would be elevation. So elevation, as we'll see, is a really important predictor variable, but it's a little bit more difficult to interpret because elevation is associated with changes in temperature, increasing elevation, decreasing temperature, but it's also associated with soil conditions. So high elevations tend to have thinner soils, less available nutrients. So when you see a species responding to elevation, it's difficult to say exactly why to create a causal relationship between the distribution of that species and these indirect variables. But like I said, they can be really useful predictors. And because remotely sensed data can be acquired easily across a large area, it can be really valuable in that sense as well. Examples that we used would be topographic wetness index. So you're looking at a topographic map here. And if you look at a particular location in this topographic model, raster cell, if you're familiar with GIS, topographic wetness index would look at what area would be draining into this particular location, what area would be contributing water to it, and what is the ability of that location to shed water. Essentially, what is its slope? And this allows you to calculate topographic wetness. It's been shown to be associated with soil moisture, soil depths, soil nutrients. And so it's associated with a lot of important ecological variables and is a really useful predictor, but difficult to interpret its causal relationships because of those multiple correlations. And then an example from our spectral data would be the normalized difference vegetation index. So this is looking at spectral reflectance, and it's associated with health and vigor of vegetation, but it can also change with chemical constituencies. For instance, deciduous species and conifer species have different responses for the NDVI. And so again, important predictor variable, but can be difficult to interpret. And so then we took these different predictors and created different types of models with them. So we created model classes with just soil variables and models using just spectral variables, models with just topographic variables. And then we created a model with both topographic and spectral variables. So that's the combination of all of our remotely sensed variables. and then a model class that we called all, and this allows any combination of our predictor variables to be used. That's kind of our benchmark for the strongest possible models that we can create. It has the greatest flexibility. And so we'll look at this all model class as kind of an example of how we constructed these models within each model class. So as I mentioned, in the all model, we're allowed to use any combination of predictor variables. And we wanted to avoid some of these stepwise model building techniques because there are some well-recognized limitations in that methodology. And so as an alternative to that, we created all the possible combinations of predictors and then looked at which model was the strongest of those combinations. So we had 23 total predictor variables in the study. We're creating a 3 predictor model. So that's 23, choose 3, results in 1,771 models. And then you look at those models and determine which one is the strongest. And so a little bit more about the actual statistical models that we're using, and then we'll move on from statistics. We have our data set here, and we have the presence and absence of species and the environmental conditions where species are present and absent. We use 5-fold cross-validation, so you break that data set into 5 parts, and you choose one part at random. And this creates a training data set and an evaluation data set. And in the training data set, you're basically estimating niche. You're looking at presence and absence and environmental conditions, estimating the niche, and then you come over to the evaluation data set and you say, here's a site with these environmental conditions. Based on our estimate of niche, do you think the species is present or absent here? And then you evaluate how successful you've been using Cohen's kappa, which accounts for some of the random chance of agreement. And to actually estimate niche, we used several statistical models and combine these using what's called an ensemble model. And ensemble models are pretty common methodology in SDMs, at least coming out of Europe, where a lot of research on SDMs has been done. And it's been shown to create more robust models. So these models perform better if you bring them to different geographic regions and apply them. And it also has been shown just to improve the predictive accuracy of models, though there's some discussion about how ensemble models are created and how appropriate they are. Once you have your ensemble model, you then evaluate how well it did with Cohen's kappa. And so now to get back to our objectives, first thing we're going to do is determine how important are soil variables in SDMs. And there's 2 factors that really determine this. The most important one is scale. And so this figure from Pearson and Dawson, 2003, kind of outlines this. At global and continental scales, it's really climate that drives the distribution of species. Once you get into regional and landscape scales, topography and land use become the dominant gradients. And then down at local, site, and micro scale, you start to get land use, soil type, and biotic interactions. And so, as I mentioned, not too many studies have looked at soil variables in SDMs. The studies that have, though, have worked at larger scales outside of which soil type is recognized to be a dominant gradient. And so our study is working right at the interface of landscape and local scales. And this makes our study uniquely suited to determine the importance of soil variables. Then another factor is what study species you're looking at. So multiple studies have shown that tree species are less sensitive to soil conditions than understory species. And the majority of these studies that have looked at soil variables in SDMs have looked at trees and shrubs. And so there's another factor that likely minimizes the importance of soil variables in SDMs. And so we're looking at understory species. And so, that combined with the scale that we're working on allows us to emphasize the potential importance of soil variables in SDMs. And so what did we find? First, I'll bring your attention to the x-axis at the bottom here. You'll see the number of predictors. So we created 2 predictor models and 3 predictor models. And we actually created 1 predictor models as well, but all of those failed. So they're not represented here. The reason we created 2 and 3 predictor models is the more predictors you use, the more observations of a species that you need. So if you have 2 predictors, you need 20 observations of a species. If you have 3 predictors, you need 30 observations of the species. For 4 and 5 predictors, you need 40 and 50 observations. And so that becomes limiting regarding which species you're able to model. You'll only be able to model the most abundant species. And we're interested in the rare species. We already lost some by these requirements of the number of observations, but we're interested to see how many variables are needed to accurately predict distributions. And so what you'll see is 2 predictor models did not perform as well as 3 predictor models. And we can look over on the left at this Landis and Koch assessment of kappa to get an idea of what these values actually mean. Two predictor models generally performed between 0.6 and 0.7, which is the low end of what's considered a substantial model. And the three predictor models more around the 0.7 region, the all three model performing up above 7.5, so kind of mid-range of substantial. And to give you a better idea of what this actually means, one of our models had a kappa value of 0.71, so right in the middle of what's considered substantial. And that's a sensitivity of 90. That means 90% of the time that our model said the species was present, it was present, and a specificity of 82.1. So 82% of the time that our model said the species was absent, it was in fact absent. And so the other thing that you notice in this figure is we have different colored boxes, and those represent our different model classes. You'll notice the green box is the spectral model, and that's the worst performing model class. In the yellowish color, the blue, and the purple, these are the soil, topo models, and topospectral models. And those perform similarly both with 2 and 3 predictor models. And then the last model class in the orange, the all model class, which allows any combination of predictors to be used, these were our best performing models. And so if you compare our all model class to our topospectral, the all model class is basically just this topospectral model class with the addition of soil variables. And so you see an improved predictive accuracy in the all model class, which suggests that the addition of soil variables to a topospectral model does improve predictive accuracy. It doesn't improve predictive accuracy by much with 2 predictors, but it is a considerable amount in the 3 predictor models. And it would be interesting to see with 4 and 5 predictors. You can notice that these model classes have greater separation in the 3 predictor model class, and it would be interesting to see if model classes are increasingly divergent as you add more predictor variables. Another thing to notice, I kind of already mentioned this, but soil models and topospectral models perform similarly. So they have similar predictive capabilities. And then for our strongest models, the 3 predictor models that allow any combination, 94% of those models contained a soil variable, 79% contained a topographic variable, and 29% contained a spectral variable. So again, spectral variables are not as important as these soil and topographic variables, and the soil and topographic variables have comparable importance to each other. Now here's a look at the individual predictor variables themselves, and you'll notice we have our 3 different predictor classes: soil, spectral, and topo. And there are different numbers of predictors in each class, which isn't ideal for comparing the predictive capability of these different classes, but it's a reflection of the different amounts of multi-collinearity in these spectral classes. So for example, our spectral class, we started off with 50 predictor variables, but there's high correlation between those predictors, and there's high variance inflation between those predictors. And so that number of predictors need to be reduced to meet some of the assumptions of our statistical models. And so the fact that soil models and topographic models have more variables is really just a reflection of the fact that there's lower multi-collinearity in those predictor classes. And so if we look at some of the specific soil variables, so you can get familiar with the naming convention here, BCA would refer to the B horizon calcium, and the OAAL would refer to the OA horizon aluminum. And so what you notice here is that the 2 most important predictor variables are B calcium and elevation. They have the highest variable importance, and they're also used in the greatest number of models. The other thing I want to bring to your attention, though, is that almost all of our predictors were used. We had 1 spectral variable that's not shown here because it wasn't used in any models. But other than that, all of our available predictors were used in at least 1 model. And in addition, of our 41 study species, no 2 species used the same exact subset of predictor variables. So this kind of highlights just how unique species are. Their niches are unique, and there's a need to have a variety of predictor variables to model them and flexible model building techniques. And so to build on this idea of flexible model building techniques, we'll look at this figure. 2 of these model types you're already familiar with. The one on the farthest left I referenced as the benchmark. These are our strongest models, 3 predictor models using any combination of predictors. We were just looking at that on the last slide. And then this remotely sensed model, again, this is just our 3 predictor model using topo and spectral models. These 2 other model types, though, these are an attempt to use a subset of predictor variables and apply that subset to all of the species. So can we successfully model all of our species using the same suite of predictor variables? And the way that we did this was we tried to choose our 3 best predictor variables. And so the universal best looks at the 3 variables with the highest variable importance. So that's elevation, BCA, and NDVI. And the universal most model looks at the, uses the 3 predictors that are used in the greatest number of models. So that's elevation, BCA, and TWI. And you can see these models do not perform as well as these other more flexible model building techniques. And in addition, a large percentage of these models actually failed, 29% for universal best and 24% for the universal most. And so this just highlights how unique species are. You can't take one subset of predictors and apply them to all species. Even if you're using something like B calcium, which is a really useful soil predictor variable, if you don't allow that flexibility, it won't even perform as well as our remotely sensed model that does have flexibility. And then the other thing I want to mention is I talked about tree species having a weaker association with soil variables than non-tree species. And we see that in our results as well. If you look at the importance of soil variables were less important for tree species in the teal there than they were for non-tree species in the red. And conversely, topographic variables were more important for tree species than they were for non-tree species. So this confirms what some other studies have found. What's interesting about this, though, is we looked at tree species in the understory. And so the other studies that have found weaker associations between trees and soils have been looking at overstory trees. And their conclusion about why that relationship exists was largely that the overstory maintains some sort of legacy effects of past disturbance, silvicultural treatments, or land management. And so that you're really picking up on that signal of kind of history rather than the signal between soils and the overstory. But because we looked at tree species in the understory, individuals that are regenerating likely under a canopy, this is suggesting that there might be more going on here, that trees actually physiologically have some sort of less association with soil conditions than the non-tree species. And to kind of build off of that, we looked at what's called the Ronquier plant life form classification system. So this is a really successful functional grouping of species, of plant species, developed in the early 1900s. And Ronquier defines species based on the location of their perinating bud during the season with adverse conditions. So that's the winter in our climate. And we had 3 different groups. We had hemicryptophytes, like this violet, that has a rhizome or a perinating bud near the soil surface, geophytes, which have a perinating bud below the soil surface, like a bulb or a culm or something like that, and then phanerophytes, which have perinating buds above the soil surface, like trees and shrubs. And so what we saw here were that soil variables were most important for hemicryptophytes, these species with perinating buds near the soil surface, and they were less important for geophytes and phanerophytes. And in addition, topographic variables were more important for geophytes and phanerophytes than they were for hemicryptophytes. So this relationship with phanerophytes, again, phanerophytes are trees and shrubs. And so this largely confirms what other studies have shown and what we looked at on the previous slide, that tree species have a weaker association with soil variables than non-tree species. But with this classification, we're kind of breaking down those non-tree species. We've got geophytes and hemicryptophytes, and it's suggesting there might be more relationships here, different ways of looking at functional groups that also kind of explain the relationships between species or functional groups and environmental conditions. And so for an overview of Objective 1, we wanted to determine the importance of soil variables in SDMs. And we found that soil variables do improve SDMs. But what's important to recognize is that remotely sensed SDMs still perform well. These are considered substantial models, and remotely sensed variables are widely available, generally for free. And, so this allows land managers and researchers the opportunity to create SDMs at kind of low cost. A lot of people have some data related to the distribution of species. And so these are doable techniques to create substantial models for predicting the distribution of species. Another point, soil variables are important, but you need the flexibility of how you're creating models. When we took B calcium, which is a really strong predictor variable, and just tried to create this standardized model and apply it to all species, that does not work well. So you need the flexibility to create different types of models. Having a wide variety of predictor variables is important. We used almost all of our predictor variables, and being able to create different combinations is important. And then also functional groups are important. So if you're designing a study and you need to know whether soil variables are going to be important, it may have to do with what species you're looking at. And people have talked about this association with trees and non-trees and soil relationships. But our results suggest that there may be even more relationships within this. And it would be interesting to look at additional functional group classifications and see how these different functional groups interact with different environmental gradients. And so objective 2, actually looking at these species that occur in the White Mountains, species that many of you are probably familiar with, and seeing what environmental gradients are driving their distributions. So this figure here is called a non-metric multidimensional scaling ordination, which sounds complicated and it looks kind of confusing, but it's important to understand because it really represents our current understanding of species and communities and how environmental gradients are associated with them. So if you look at these 2 points here that I just put arrows to, these 2 points and every other point on this figure represents a plot that we went to. And plots that are close to each other have similar species compositions to each other. So these 2 plots maybe have a lot of beaked hazelnut, and elderberry and foam flower. They have similar species compositions. And the color of the plot represents the community type. And so these are pink plots and they represent semi-rich communities. And so you would expect plots that are close to each other that have similar species compositions to be classified as the same community. So it makes sense for points that are close to each other to be classified as the same community or have the same color as each other. Now you'll also notice that out of the center of this figure, there are these vectors coming out with environmental gradients labeled on them. And so pointing up towards the 2 plots that we've been looking at, you see OAPH and BCA. This is suggesting that these plots are associated with high OAPH and high B calcium. And so if we look at the dominant gradients on this ordination, we see elevation is one of the dominant gradients, represented by elevation on one side and NDVI on the other side. NDVI tends to be high where there are more deciduous species, and deciduous species tend to be at lower elevations. So we're looking at an elevational gradient here. And then we have a fertility gradient. Fertility tends to be high where pH is high, and fertility tends to be high where calcium is high. Calcium is what's called a base cation, and base cations tend to be high at high fertility. Then the opposite side has acid cations, and aluminum is an acid cation. It's not shown here, but we'll see it on the next slide. So aluminum is associated with low fertility, and also associated with low fertility is a high carbon to nitrogen ratio, which we see on the opposite side of this gradient, BCN standing for B carbon to nitrogen ratio. So these are the 2 dominant gradients, elevation and fertility. And BC to N is really the variable that represents this fertility gradient. Elevation and B carbon to nitrogen ratio have an R squared value, both of them, of 0.55 with this ordination. They're the strongest variables. They're really driving the community composition here, and they have very similar importance based on this ordination. And so if we look at this species up here, these 6 letters codes represent species. So Athang references ladyfern. And so I mentioned that this ordination kind of represents our current knowledge about species and environmental gradients. So what we would interpret about ladyfern based on this ordination is that it tends to kind of cluster around some of these pinker plots like semi-rich and rich plots. And the environmental conditions it's associated tend to be higher OAPH, higher B calcium, and higher TWI mean. So very correlative kind of understandings of these things. And what we're not able to do is say, okay, lady fern's associated with B calcium, but how high does B calcium need to be before we start to actually see lady fern in a plot? And those types of more quantitative relationships are what species distribution models allow us to do. So one of the species that we know about as much as any other species about for our area, because it's iconic for the region and it has economic importance, is sugar maple. And Haynes, who wrote the flora for this region, described the habitat as mesic forests, including uplands, rocky slopes, and high terrace floodplains. And Sperduto and Nichols, who wrote the New Hampshire Community Classification System, talk more about sugar maple and how it's associated with higher fertility sites. So these sites with high pH and high base cations. So here's our model for sugar maple. It had a kappa value of 0.76, a sensitivity of 82.9. So 82.9% of the time that the model said the species was present, it was present. Specificity of 100. So 100% of the time that the model said the species was absent, it was absent. And if you look down at the bottom left, it describes the variable importance. The most important variable here is elevation by far. And we see a precipitous decline in the probability of occurrence of sugar maple at elevations around 750 meters. The second most important variable is B aluminum, which I talked about being associated with low fertility conditions. So as B aluminum increases, the probability of occurrence of sugar maple decreases. So this is describing the same trend that Sperduto and Nichols talked about, and many other researchers have described, that sugar maple is associated with higher fertility conditions. But at least when you look at the study area that we looked at with a kind of a larger elevational gradient, that's really overshadowed by this relationship with elevation. And so here's a description that's kind of unique, pretty quantitative description coming out of the US Forest Service Southern Research Station. And we don't have many relationships like this. The reason we have this kind of relationship is that we know a lot about sugar maple, and a lot of people have studied elevation. So we know more about this than species responses to soil chemistry, for instance. And what this excerpt describes is that around elevations of 760 meters, sugar maple starts to really disappear quickly. And that's exactly what we see in this model as well. So that provides some confirmation for what we've seen. And so we know a lot about sugar maple, but for the majority of the species that we modeled, we know much less about. For instance, painted trillium. This is a species that's common in the White Mountains. You see it down in southern New England as well. But we have much less of an understanding of how it associates with soil conditions. So Haynes described its habitat as mesic deciduous forests, less frequently in mixed evergreen deciduous forests. And Sperduto and Nichols didn't describe it in association with a particular community type. Sperduto and Nichols was a community classification guide, so they're describing particular communities, and when species are strongly associated with communities, they describe those relationships. But their intention wasn't to describe each species and what environmental conditions they appear in. So for painted trillium, what we saw is a kappa value of 0.71, sensitivity of 90, specificity of 82.1. And the most important variable is B aluminum. And we see increasing probability of occurrence of painted trillium with increasing B aluminum, suggesting this species is associated with low fertility sites. The second most important variable was B calcium. And interestingly, we see a momentary increase in the probability of occurrence of painted trillium with increasing B calcium. But the overwhelming trend is as B calcium increases, the probability of occurrence of painted trillium decreases. And that's more what you would expect because aluminum and calcium are antagonistic with each other. And so together, those two really suggest that at high fertility sites, painted trillium is not present. This is more of a low fertility site species. And so I'm going to change gears a little bit and talk about Ellenberg indicator values. So this is a system that's designed to use plant species to indicate environmental conditions. It was established in Central Europe in the early 90s, and it's been widely used for 30 years in hundreds of studies. It's used commonly in species distribution model literature, and the system's designed based on expert knowledge, assigning ordinal values to species for different environmental gradients. So an example is one of their gradients is pH. And so if we were to do this with our species, we saw painted trillium as kind of a lower fertility species, maybe associated with more acidic conditions, and we could assign it a pH value of 3. Whereas sugar maple is a more high fertility species associated with likely higher pH, and we could assign it a value of 7. And so this is a bit whimsical. You know, there's much more research that goes into assigning these Ellenberg indicator values. But I'd also just like to say, I think expert knowledge is really important. A lot of the relationships that we are seeing between species and the environment in New England have been described with expert knowledge, you know, somewhat anecdotally for a long time. And more recent research with chemical analyses and more complex statistical analyses have largely confirmed the relationships that have been described for quite a while. And so Ellenberg indicator values look at 7 gradients: pH, fertility, soil moisture, salinity, continentality, light, and temperature. And if you bring your eyes down to the bottom of this slide here, we see this species, Atherium felix femina, which is lady fern. The scientific name's been changed to Atherium angustum, but we can see there's values assigned for pH and fertility that I've drawn arrows to. So a value of 5 for pH and 6 for fertility on a 1 to 9 scale. So this is mid to mid-high pH and fertility species. And above that, you can see our response curves for these species. We see increasing probability of occurrence of lady fern with increasing B calcium, suggesting this species is associated with high fertility sites. And that's something that Sperduto and Nichols also described. I just want to kind of compare and contrast this Ellenberg indicator value system and some of these more quantitative responses. You know, the Ellenberg indicator values have a bit more of a kind of subjective nature to them because they're assigned using expert knowledge. And they're also relative to the study area that you're working in and the other species that you're looking at. Whereas these more quantitative responses can allow us to say things like, the probability of seeing lady fern is quite low below, you know, concentrations of B calcium below 200 parts per million. And so I think there's a potential for using these types of relationships that we've uncovered to create more quantitative assessments of environmental conditions. But if not that, it at least provides some empirical evidence to back up a system like Ellenberg indicator values, which has been really successful. And I think it would be useful to implement a similar system in this region. So an objective 2 overview, we're interested in looking at these species and what environmental gradients drive their distributions. And we saw that elevation and fertility are really the 2 gradients that are driving distributions of these species. And we saw that in multiple places, both in objective 2 and in objective 1. In objective 1, we saw that, you know, our strongest models were kind of dominated by soil variables and topographic variables. And when we looked at the specific variables that were dominating, it was B calcium, which is associated with fertility and elevation. And another thing to notice, I just think it's interesting that, you know, we looked at a decent elevational gradient going from about 300 meters up to 1200 meters, and to see elevation having comparable importance to see fertility having a comparable importance to elevation is pretty cool. Another thing we saw, you know, some of these models confirm relationships that we're aware of, but other models, in the vast majority of these models, are describing kind of new relationships between species and environmental gradients that haven't really been described before. And so, you know, they should be treated with caution, but it also kind of uncovers potentially some new relationships that we're not aware of. And then I think this develops a potential for developing a similar system to Ellenberg indicator values. So this has been a really successful system in Europe, and there's no corollary system in North America, you know, and I think we could at least use this to establish some sort of ordinal expert knowledge-based system, if not to go beyond and actually predict some sort of kind of quantitative assessments of environmental conditions. And so with that, some closing thoughts. I think SDMs are really practical for both land managers and researchers. You know, a lot of people have data related to the distribution of species, and that combined with remotely sensed data, which in general is freely available, can start to make some really strong species distribution models that are important for land management decisions, but also for kind of understanding the ecology and evolutionary history of these species. And then in addition, I think, the quantification of niche in general is really powerful. People have been talking about niche in an academic sense for over 100 years. And it's really just in the past 10 to 20 years that we've had all the tools to really start quantifying niche the way that we are, especially including soil chemistry and some of these GIS related techniques and statistical techniques. And so, the majority of our species, we don't know these kind of quantitative assessments of niche. And just like estimates of niche are kind of fundamental to species distribution models, I think they're kind of fundamental to us understanding these species. And once we understand the niche of these species better, there's a lot of different directions we can go in to apply that in a management and research perspectives. So with that, I'd like to thank the U.S. Forest Service, the NRCS, and the University of New Hampshire, my committee, Dr. Mark Ducey, Professor of Forest Biometrics at University of New Hampshire, Dr. Tom Lee, Professor Emeritus at the University of New Hampshire, and Dr. Rich Hallett of the U.S. Forest Service. And then, as Mark mentioned, Robert Coulter and Olivia Frazier from the Forest Service were invaluable in getting this project started. This started off as Robert Coulter or Andy Coulter's PhD research, and a lot of people did a lot of work to get this off the ground. And then to Emily P. Shea, who I had the pleasure of working with in the field, and the other past field crews who helped collect all the data. And then to my family, my mom, Karen, my dad, Gene, and then my sisters, Casey and Sarah. And you can contact me at nateroe@gmail.com. I'd be happy to send people, we have 39 other species with response curves. And if you're interested in seeing those relationships, I'd be happy to send those out. I sent some people an email directly, and for those people I can send that out. If I didn't, just send me an email and I can get those to you if it's of interest. And so there's my references if you want to check the MLA format. And yeah, if anyone has any questions, I'd be excited to hear them.
Great. We do have time for some questions from the general audience. We'll have the committee hold our tongues because we'll have a chance to have a go at Nathan here when the rest of the audience has left. We've got more than 40 people on Zoom, so I'm going to suggest that the easiest technique might be if you enter your questions in chat, and then Nathan can take them more or less in the order that they come. And I can try to help moderate that as well if I can see the chat in my screen. There we go. So questions from the audience.
All right, I see a question from Grant. Based on how robust your soil model, do you think the difference between one and the kappa and specificity values in your models are due to hyper local biological interactions such as facilitation and competition? Grant, do you want to unmute yourself and elaborate a little bit on that?
Yeah, sure. I didn't maybe know how to exactly phrase it because I don't know modeling as much, but you're capping your specificity values in these models. There was a difference between on how well they could predict. Is the difference where the models are missing, is that based off of those hyperlocal biological interactions that you showed? They're on the 1 meter to half a meter scale.
Sure. Yeah. So that's a good question. Yeah, where the models didn't perform as well as others is really interesting. What could potentially be causing that? I think one immediate thing that I would think of is this dispersal ability, how well dispersed species are. I mentioned that being one of the assumptions. And so when species don't appear where you would think that they should be in an area that kind of seems like their niche, it does two things. It messes up your estimate of the niche of species, but it also messes up your predictions. And so your accuracy in predicting gets messed up. So, that's one factor that could definitely affect it. And different species, particularly species that are like ant dispersed, some of these things, some species are well known in New England to not be very well dispersed. And so that's a violation of one of the assumptions of SDMs. And that could certainly contribute to those factors. But like you're saying, biotic interactions between species, that's something that that we didn't model. And so I recognize that we didn't fully include every variable that's important to the niche of these species. So that's another violation of assumption. But we're more interested in seeing, just how important are soil variables and how does that compare to our ability to predict these things using remotely sensed variables. So those are all good points. Biotic interactions, things like that could certainly be reasons that certain species didn't model as well as others. Let's see here. Ben, this is really impressive. Does probability of occurrence have any bearing on abundance? Oh, good question. Also, second question, in a presence absence model, how do you choose a spatial scale for your model? So presence absence is kind of like, that's what's generally used in species distribution model literature. But one of the things people are talking about is that, including abundance is really the next step. There are several next steps, but that's one of them. It really improves your ability to describe species response to environmental gradients. And so people are creating SDMs right now with abundance, and probability of occurrence probably does have a relationship with abundance. If it predicts really high probability of occurrence, I'd be willing to guess that it's probably abundant in that area if the model's performing well. But yeah, things that predict abundance are coming out, and that methodology is just kind of a little bit more primitive right now, but it's certainly coming. And then how do you choose a spatial scale? So we chose a spatial scale just trying to use the finest scale that we had. Obviously our soils data is kind of like, it's point data, so it's very fine resolution, and our LiDAR data was fine resolution. And our spectral data was not as fine. And so we're kind of combining, there's a little bit of combining different spatial scales in a way that works. But for fine scale studies like this, it's ideal to be working at fine spatial scales, because in particular, soil variables change a lot in small scales. Within a 10-meter radius plot, soil conditions can change quite a bit. And that's another factor that is somewhat problematic in determining relationships between species and soils.
Cool, thanks.
Yeah. Russ, what date did you use for NDVI? Seems in New England forest that canopy is quite dense, so cannot really. Okay, yeah, that's it. So Russ is saying that NDVI, that canopy is really dense in New England forest, and so actually seeing down to the understory would be difficult. That's a good point. And so NDVI we used in the summer. It was a date. All of our spectral data came from a date where there was almost no cloud cover. And for sure, I agree that you're likely not actually picking up the understory species themselves. But I guess, in the sense that NDVI can pick up on some differences in chemical constituents and maybe things associated with kind of fertility, that hopefully the overstory might be kind of a representation, where you get more sugar maple in the overstory. If you're able to pick that kind of thing up, that might be kind of indicative of what's in the understory as well. Because there are, of course, associations between overstory and understory. Brent Powers, wondering if you looked at the effect of agriculture on herb and shrub species distributions. So that's a really good question. It kind of fits into, I think it was the question that Ben asked, things about distribution. So past agricultural use in New England is well recognized to have extirpated plants from these regions. And so particularly these ant dispersed species is really slow process for them to get back into these areas. And so land management history is a little fuzzy in the White Mountains. I'm not aware that any of the areas that we worked in had past agricultural use that might have that level of impact on the distribution of species. But if you're working in an area where you did have known past agricultural use, several studies have shown that that really impacts the distribution of species, that heavy disturbance in the past. Michael Thompson, do you know if something similar could be done with interspecific interactions rather than environmental variables? Well, I know interspecific interactions are something that's kind of falls into this biotic interaction, which at small scales, those are really important. Yeah, I think it might be tough if I'm thinking about the question correctly. You know, that might require an understanding of what species are kind of already at these sites in some sense, but there's certainly associations between different species, you know, and where one species occurs, another species tends to occur. So there's certainly relationships there that I think could be explored. And yeah, that's a component that ideally is incorporated into SDMs is, you know, there are these competitive and facilitative interactions between species, and it's important to account for those. And that is an important factor in defining the niche of species. So, you know, I think you could potentially do that. You know, it'd be ideal to have some environmental conditions as well, but determining associations between variables, I think would be between species would be important. Russ said, did you think about other spectral variables besides NDVI? Yeah, so we looked at NDWI, and then I looked at a variable called mcari -savi, which is a variable that I'm actually not too familiar with. And the reason that I looked at it was based on one of my NMDS ordinations. It seemed to have kind of a unique property where it was pointing in an association with kind of a fertility gradient. And what I saw most of my spectral variables doing is really just associating with kind of a elevational gradient. It seemed like kind of this difference between deciduous and conifer species was dominating a lot of these spectral variables. So I included this mcari -savi variable, and that's the variable that ended up not being used in any models. So it looked interesting from my NMDS, but turned out to not be very important. But it would have been nice to include more of these spectral variables. But as I mentioned, the multicollinearity was just so high that we weren't able to include many of them. Jeff Garnis says, how much does the range of variation in each of your variables or variable grouping drive their predictive power? Nice. You surely have decent elevational variation, but I'm less sure about variation in soils. Yeah, so that's a really good point. So what's ideal to have is a full gradient of the distribution of kind of these environmental variables. So go from really low pH to really high pH, and that allows you to see kind of what Whitaker described, these species responses that are kind of bell-shaped curves. And so you don't see a lot of bell-shaped curves in our responses because we're not representing the full gradient of these environmental variables. And so, you know, it's definitely a limitation in defining the niche of these species. We're kind of, for a lot of them, maybe defining one side of the niche, but we don't know kind of where the upper threshold is. Like, we're not finding upper threshold of fertility in these sites. And so that's certainly a limitation. And, you know, you need to design your study likely over broader geographic regions to kind of explore that. And that's one limitation of creating SDMs for small regional areas, is that you're not able to represent those full environmental gradients. Justin, if you're aware of Mark Anderson's climate resiliency work, one of the key underpinnings is a paper that Anderson and Ferry published in 2001 showing that the 4 drivers of species in the Northeast states are the area of calcareous bedrock, elevation, geologic variety, and latitude. Your work seems to validate. Yeah, I would definitely agree. That seems to be consistent. Yeah, I thought it was really cool that, you know, over an elevational gradient going from, you know, 250 or 300 meter elevation up to 1200, seeing that that fertility was just as dominant a gradient as elevation was. And I think that you likely would not see that for overstory species. I think it's likely that understory species have been recognized to be more associated with soil fertility. And I think that really brought out the importance of that gradient. Andy Coulter says, thinking how to move forward with TEUI modeling, which is terrestrial ecological units. So that's kind of where this project started, was Andy trying to define community types using some of these remote sensing technologies. He says, we use New Hampshire community types to create a predictive TEUI model, but should we be using some type of understory classification instead to create a new TEUI prototype surface? One response to that I think is interesting, in species distribution model literature, a lot of people talk about the need to really model species individually rather than communities. And we know that kind of novel communities are rising as a result of, global environmental change in general. And so I think that, yeah, understanding species individually is really interesting. And then you can combine those to kind of create an understanding of what communities are there. But yeah, I think, this understory classification, Sperduto and Nichols includes understory a good bit in their classifications. And one thing that I kind of just anecdotally noticed was that there are community classifications that seem to incorporate more understory species. I found to kind of respond more in the intuitive ways that I expected them to understand with some of these statistical models. And so a lot of kind of these more rich community types, semi-rich and rich mesic forest, those were kind of ordinating in the places where I expected them to ordinate. And I think the addition of understory species, because they're referenced more in those rich communities than the other communities, I think that really helps improve the classification. So I think the more that you're including understory in classifications of forests is really helpful. One thing to keep in mind is that 80% of the species richness in eastern forests is the understory. So if you're ignoring understory, you're ignoring 80% of your species. Olivia Fraser says, given the role of soil chemistry as a predictor, how feasible is it to model understory species across the White Mountain National Forest? And how many plots to what soil depth do you think would need to be accomplished for a forest-wide model? So I think, if you're going to create forest-wide models, one of the things we saw is that remotely sensed models performed well. And I think one of the reasons that they performed well is they are able to account for soil conditions. Like I talked about topographic wetness and some of these spectral variables, they're correlated with soil conditions, and we're not going to be able to model, or we're not going to be able to sample soil conditions across the whole region, across the whole national forest. And so it's going to require improving our understanding of the relationships between some of these indirect variables and things like soil chemistry, if we're going to do that. And I know, Olivia, you've thought about this a lot, but yeah, kind of further exploring some of these remotely sensed variables, and can we predict some sort of fertility assessment using these remotely sensed variables? I think that would be the key. And yet, we had a moderate sample size here with a couple hundred plots, but yeah, it seemed like from what I was seeing, the B horizon is really the kicker in these. So that's what you really need soils from. And I think, another thing to notice, and I talked to Scott Bailey about this, pH and base cations and fertility don't have that strong of a correlation in these acidic forest soils that we're looking at. So a lot of studies have looked kind of just grabbing pH, and pH is a pretty good predictor of soil fertility, but it's not always great. Sometimes base cations are high in kind of acidic sites. And so, that makes it a little more complicated because getting base cation concentrations is more intensive soil chemical analyses than it is just getting pH. So I think, yeah, it'd be pretty tough to get enough soil chemical data across any large area without using remote sensing. Cool. I'm not seeing any other questions unless I'm missing something, but.
Yeah. Any last questions out there before we conclude the public portion of the defense? Oh, good. There's one.
Okay. One from my mom. It seems you didn't encounter many introduced invasive species in your plots. What do you think will happen as invasives increase? Yeah, so one thing that these models can be useful for is kind of predicting, how amenable areas are for invasive species. So they can kind of give you an idea, is this suitable habitat for invasive species? Even if they're not here, where might they be headed? But yeah, up in the White Mountains, we're mostly working under full canopy forests, and you don't tend to see a lot of invasives in those less disturbed areas. But yeah, as they come in, it's definitely something to consider. And I think, it's an interesting application of using SDMs to kind of predict, where might these species be headed. But yeah, we didn't encounter too many. And then, yeah, a couple questions about, will the recording be available? I hope so. Zoom is supposedly recording it right now, and hopefully that'll all work well. So yeah, I'll certainly make that downloadable. If you didn't get an email directly from me and you want additional things like species response curves or the recording, email me at nateroe@gmail.com, which is right there. Yeah. Yep. Okay, I've got one question maybe from Dan Sperduto, which would be interesting. Your work suggests that understory plants are a good way to quantify conditions across the landscape. Maybe we can use specific observations across the landscape we already have to contribute to models. Yeah, so I think it's really, really cool. I mean, a lot of people have information about distributions of species, and that combined with remotely sensed variables, I think could make some cool, if people were kind of collaborating on getting data together, I think that could really make more robust models. But yeah, and then in terms of estimating the niche, what a lot of people have done with these Ellenberg indicator values is going back to kind of old data sets. And now that they have kind of better idea of how to use these Ellenberg indicator values, they can go back and say, oh, this plot that we had here, based on the species composition and the abundance of these species, we would kind of, predict environmental conditions to be like this. And it kind of opens up a new possibility for reanalyzing some of these data sets. Adele, you mentioned disturbance as a direct variable. How might you approach developing a model for a community or species that is ostensibly fire dependent? Oh, that's a good question. Yeah. Yeah, I think, in a fire dominated ecosystem, it would be really important to be including disturbance in the definition of these species niche. We tried to sample sites that didn't have recent disturbance, nothing within like 50 years. And so and another thing is understory has been shown to respond more quickly to disturbance, getting back to similar species compositions in about 20 years after disturbance. And so if you had a community that's burning regularly, I think that'd be really important to include. And you'd certainly need, time to the last fire to be included in your model. And I think, out west, that those are probably things that they have fairly well documented is, when the last fire took place. And, you might need to include fire severity as well if you're going to make effective models, because that's certainly going to influence the composition of species in those sites. But yeah, I think that could be really interesting to include fire.
Great. Well, we should probably move on so that we don't keep Nathan until midnight tonight, grilling him behind electronically closed doors. So thank you everybody for coming. I wish that this was a normal defense and we could all walk around the room and say hi and mingle for a bit. We'll do that some other time for sure. But thanks everybody for coming. Thank you, Nathan. And we will let everybody sign off, and then in a, let's say, 5 or 10 minutes space, we'll reconvene the committee with Nathan for the closed door portion of the defense. So thank you.